Hi everyone. Today I'm going to present some of the results of my master thesis project in this talk titled Light Orientation Affects the Intensity of Courtship Behavior in the Jigwood Splitfin Xenotoca variata. Any animal behavior carries with it costs and benefits. So, in a decision-making process, an animal searches for optimize their behavior, understanding by optimization the process in which an animal solves the compromise between costs and benefits of a behavior for increasing or decreasing the consequences of a decision in order to get the maximum net benefits that a behavior can offer in a particular time and context. But what is taken into account in the decision-making process? First, signals that are behavior or structures that an animal exhibits in order to transmit a message and modify the behavior of a receptor. Then the environment, because it's where a signal is transmitted and is not the same to send a signal in the water or in the air, for example. And finally, motivation, that refers the internal immediate mechanisms that trigger a behavior depending on the needs of an animal. All these elements together are going to establish a hierarchical organization of the behavior. Within the signals that we can observe in nature, we have the visual signals, such as the iridescent structures in the integument of some animals. Iridescence is a structural color characterized by presenting directionality. Directionality is the color change with the angle of observation or with the angle of incident light. So, it's evident that there is a strong relationship between iridescence and ambient light. It could be common to observe iridescent structures in animals inhabiting low-light aquatic environments because iridescence could facilitate the transmission of signals and also could be useful for modulate the conspicuity of animal depending on the context and depending on the ambient light. An example of an animal possessing iridescence is a freshwater fish, Xenotoca variata. Xenotoca variata is a fish endemic of Mexico belonging to the family Godeidae. Males of this species lack of gonopodium. Instead, they have spermatopodium, that there is a modification of the rays of the anal fin that males use for copulating with females. Males of this species exhibit the iridescence of their scales for attracting females and copulate. It has been reported that females prefer to mate with males that have more iridescent scales, but these same males are also preferred by the Thamnophis melanogaster snakes, a potential predator. So, possessing iridescent scales represent both costs and benefits for males of Xenotoca variata. So they should be developed strategies to exploit iridescence depending on the context. For example, optimizing their courtship behavior depending on ambient light. Based on this information, I proposed the next research question. How do Xenotoca variata males optimize courtship behavior depending on ambient light? I hypothesized that males can obtain maximum net benefits from the iridescence of their scales. If this hypothesis were true, I expected that males of Xenotoca variata show more courtship displays and spent more time close to females when the incident light on water was optimal. To test these predictions, I carried out a 4x4 factorial experimental design. I tested the effect of two principal factors on the courtship behavior of Xenotoca variata, each one with 4 levels. By one hand, the incident angle of light, with 15, 30, 45, and 60 degrees, and by the other hand, the position of the light source, with north, south, east, and west positions. In this way, I have a total of 16 different treatments that I apply randomly along 4 consecutive days. Due to the I was not able to apply at the same time the 16 treatments, I considered the week as a blocking criterion in the statistical analysis. And in addition, I performed 4 replicates of each treatment to test also the possible effect of the interaction of the main factors on the courtship behavior of Xenotoca variata. I locate the lamp at the position and incident angle corresponding to each treatment. I introduced one male and one female in the fish tank, and they was surrounded by a PVC tube for preventing the contact between them during the acclimation process. After 5 minutes, I released the males in the fish tank and the female inside a glass cylinder that was located on the center of the fish tank. This glass cylinder was surrounded by a PVC tube to continue preventing the contact between males and females during this second period of acclimation. After 15 minutes, I removed the PVC tube and I started with the video recording. I video recorded the behavior of this fish during 20 minutes to register after the number of courtship displays that males exhibit to the females, and also the time that males spend close to female inside this critical zone around the glass cylinder located on the center of the fish tank. In this way, the dependent variables of my study were the frequency of courtship displays and the time that males spend close to females inside that critical zone. The independent variables were the position of the light source and the angle of incident light. And finally, I considered 3 covariates in the statistical analysis: the water temperature, the difference in standard length between males and females, and the number of iridescent scales of males. After the data collection and the statistical analysis, I obtained the next results. First, according to the time that males spend close to females, there was no effect of ambient light on the variation of this data. The minimum adequate model that explained the variation of the time that males spend close to females include just the difference in the standard length between males and females and the iridescent scales of the males. We can observe in this figure, in the x-axis, the ratio between the standard length of the males and the total standard length that corresponded to the male standard length plus the female standard length. So, smaller males spend more time close to larger females, but this time diminished when the body size was similar between males and females, and was even lower when males were larger than females. And these results could be explained because there is a relationship between the female body size and their fecundity. So males could prefer spend more time close to larger females because they represent an opportunity to have more offspring. And regarding to the number of iridescent scales, we can observe a tendency of males with more iridescent scales to spend more time close to females. And this could be explained because, according to Salazar Bautista, males of Xenotoca variata that have more iridescent scales are also the dominant males inside the population. And these dominant males are characterized by horde resources. So in this case, males could be spending more time close to females when they have more iridescent scales because they are showing their ability to hoard resources, in this case, females. By the other hand, according with the frequency of courtship displays that males exhibit to females, the minimum adequate model that explained the variation of this data include as fixed effects the position of the light source and the angle of incident light, and as random effects the blocking criterion and the water temperature. We can observe in this figure that males courted more the females when light fall on water at 15 and 30 degrees compared to 45 and 60 degrees. So under this angle of light, males could be increasing their conspicuity for being more attractive to females. And this is similar at what happens with the males of Pavo cristatus and Volatinia jacarina, who have also iridescent feathers that exhibit to females during courtship. According with the Snell's law, when light falls on water at 15 and 30 degrees, it falls on the iridescent scales of the fish at 40.6 and 46.5 degrees. And if under these light conditions, males are increasing their conspicuity for being more attractive for the females, what about the exposure to predators? It has been reported that thamnophid snakes use visual cues that contrast with the background for locate their prey and for direct their attacks. But despite that they have color visions, they have only a few UV receptors on the retina that are useful just for spatial location of prey, but not for forming accurate images of them. By the other hand, when light falls on the iridescent scales at 45 degrees, the iridescent scales produce the minimum reflectance, the minimum brightness, and a reflectance peak between 300 and 400 nanometers that correspond to the UV spectrum of the light. So, under these particular light conditions, males could be producing UV signals during courtship for attracting females, but that are not visible for predators, and also producing the minimum brightness required to attract a potential mate, but not a predator, optimizing in this way their courtship behavior. Furthermore, during courtship, males could be moving in a particular way their body for catching the light and for produce these signals that appear and disappear for attract a potential mate, but for distract a predator. So, during courtship, Xenotoca variata males could be producing these temporal patterns of colorations that is very similar at what happened with the males of the butterfly Hippolymus bolina. The males of these species of butterflies have these iridescent UV spots on the wings that are visible just in a range of 20 degrees. So every time that they flap their wings during courtship, they are sending to the females these signals that appears and disappears. Now, according with the position of the light source, males courted more females when light came from the west than from the north and south. So, males could be orienting themselves according to the position of the sun during courtship. And perhaps they could have the ability to detect the magnetic field, like it happens with the salmon on Corinchus nerca. In summary, males prefer to court females more when the incident angle of light on water is 15 and 30 degrees and when the light comes from the west. So, light orientation affected the intensity of males' courtship, and with these results, we can conclude that Xenotoca variata males, in fact, optimize their courtship behavior depending on ambient light. Thank you so much for attending my presentation, and please, if you have any comment or any question, don't hesitate to write me. Thank you.